I'll continue with my introduction. Mostly, they have been a dynamic duo for 25 years, researching together, sometimes on the same expeditions, looking for animals in the forests of their home country, Brazil. And they really have brought this great expertise and enthusiasm to our students here at Humboldt. And I'm really excited that they can share their stories here with the community of their observations and their discoveries and their conservation of wildlife around the world. Thank you so much for being with us.
Thank you, everybody, for coming, and thank you, Michaela and Ruth, for the invitation. And when you sent the invitation for a joint talk, I started to discuss what are we going to talk about. And it just makes sense that we share some of the experiences that we've had together. So I was searching for the very first time we did field work together. So this is from 2006, and this is the very first photo that I have. We did field work back in 2004, I think the first time, but this is the first one that I have a shot of. So this is Silvia taking that picture right there. Collecting tadpoles with my dad's spaghetti filtering thingy. So this is 20 years ago. So we've been really doing field work together for over 20 years. We did meet at the university when we were undergraduate students. So this is us presenting a poster in biochemistry, not my favorite subject. In 2005, and this is us doing consultant jobs in 2008 as master students. And we did go to New York to do our PhDs, and we both worked at the American Museum of Natural History. So we've been doing work together for all this time, and we started dating in 2003, so that's 22 years. We've been married since 2010, so that's a lot of years before we started our PhD, and now we have two wonderful kids that will be in the presentation a little bit later. 2 slides as usual. So this is me. I'm a herpetologist and wildlife photographer. I do combine some of my photographic skills into my work sometimes, but most of my expertise is in the study of amphibians and reptiles, mostly amphibians, mostly frogs, mostly Amazonian frogs. And these are some of the frog species that I have named over time. This is a group that I studied for my PhD. This is a species named after Ozzy Osbourne. You might have seen some on the news. This was out of the news when Ozzy was pronounced dead a few months ago. There.
Yeah. Oh, that's Hi, everyone. Thanks for coming. My turn. So this is me. I am a mammalogist, as you already know. Here I have illustrated some of the mammals that I have worked in the past. I mostly work with taxonomy, systematics, and evolution of small mammals. The first way.
You have to point here.
Oh, sorry. Do I have the pointer like thing? No, it's fine. Okay. This first guy here, the rodent, was very, very first group of mammals that I have worked with. And I got to name this species when I was still an undergrad student at the end of my undergrad. That's from Atlantic Forest in Brazil. And then after that, I jumped to the Amazon for my master's and I never got out. So this guy was named from the Brazilian Amazon in 2017, is one of the opossum species that I have worked with. And then I had to pass here. So during this time, the 20-something years that we started our academic paths, we have developed passion and very strong interest in several shared areas, myself and Pedro, including fieldwork and exploration, which is the focus of what we're going to talk today, systematics and taxonomy, like discovering and naming species, who we have in terms of species, where they are, and the history of the region of this diversity, so the evolutionary biology and how they are related to each other. And how this knowledge that we gather with our science can also be used for some conservation purpose, right? So we've been in several places doing our, fieldwork projects, expeditions. And today I want to share with you a little bit more about 3 of those shared expeditions where we have been together. The one is at Abiseu, Peru. I call it Rio Abiseu, which is the national park in northern Peru. The mission of this expedition, the major task was to find the very mysterious species of squirrels. I'm going to talk about that soon. The second one here in the map, it's in Cachoeirão, Brazil. That's already eastern Amazonia. That was a collaborative grant with a multi-task team that had not just science purpose, but also science outreach and kind of like communicating with the general people, like community in general, about importance of understanding diversity in Amazonia. And specifically, we were like focusing on like night creatures. And this third one, which is also there on the eastern Amazonia, Marajó, is a current project that we have started more recently. We started that in 2024, and the expedition happened, the field work happened in 2025, but that's ongoing. So, first one, that's Peru, Parque Nacional de Rio Abiseu. That's the name of the National Forest Park that we were at. This was a expedition that was supported by a National Geographic grant, and the 2 of us were like having the idea together about like why we would like to go there. And I'm going to tell you like how the story, like why Parque Nacional do Rio Abiseu. So I was studying my postdoc, squirrels, and I wanted to build a tree of life with our American 3 squirrels. That's a little, too much. But then we actually were really successful in sampling all these squirrels to understand the genetic diversity of the squirrels and to build a tree of life, which was a base for our other type of studies that we could do with that. But we were missing this one very mysterious squirrel, which was known from a single specimen collected over 30 years ago at Parque Nacional de Rio Abiseu. And that was a species of squirrel that had been like in the literature, had been mentioned as a possibly new species, unnamed species. And we didn't have that squirrel in our tree of squirrels. And I was like, we need to get this, right? So that's basically why Parque Nacional de Rio Abiseu. But the place is not just a nice place to visit for the squirrel. It's also a very unique place. This is a UNESCO site, which is considered a World Heritage Site with unique importance in cultural and natural aspects. So there are only 2 sites like that in Peru. And the other one, maybe someone can guess, Machu Picchu. Yes, this is the second. So that's really not as known as Machu Picchu, but it shares like the importance, right? And so with the story of going to a very remote place, chasing a very mysterious squirrel, but also with the story that the place was really understudied for small vertebrates. We wrote that purpose, and National Geographic gave us an explorer's grant to pay for the expedition. And then, and by the way, just like putting the context of the photos, this is a view of the park with one of the archaeological sites, and over there a picture in another archaeological site, and the soles, Peruvian soles, the money in Peru, is illustrated, the 100 bill is illustrated with one of these archaeological sites from Abiseu. Which is from the pre-Incan culture known as Chachapoyas, right? Yes.
For those of you that know Spanish, this site right here is known as Los Pinchudos.
Okay, no more. Yeah. No more. And okay, so we wanted to get to this really beautiful place, but then we got to go through other, you have to take a route. And this was our route. And this is the truck. So you have an idea. Okay, this was just part of what we faced. I remember really well when we were driving in our little, car. And then it's, the mistress is like, don't look to the right, just like look in the front. No space for like sides looking. And Pedro turned back and he looked at me and he said something like, I don't remember the specific word, but something like, I'm never gonna do this again in my life. If we don't die here, I'm not coming back. So I remember these really well. And I remember also some little inconveniences that happened. So we were in a car at the beginning, the whole team in one big car, which of course fried in 3 hours after the start of the road. And then we had to change all of our equipment to a couple of other cars, which took maybe 8 hours to get there. Okay, so we were waiting, unloaded, and then waiting in the street in the road for several hours to get the cars, and then load our things again. And then we got some, flat tires. This is a biologist / mechanic helping, right? Felipe, he was also here. That's like his face when he opened the car that was fried, and he just like, oh, that's not gonna work. So we had to call the other cars and they came, but it took a long time. So a road trip that was supposed to be, I think, 8 to 12 hours took 2 days. Well, eventually we got there, which is the good news. And after we got at the park, we had to walk for 2 days, basically. And this involved helpers from the local community, also mules to carry the field gear. So some photos of these. Walking, more walking, right? More walking and pretty views. This was like going up and down. So really pretty, but also very altitude sick. For the Brazilians, the Peruvians were doing great, but the Brazilians were like, okay, wait a little bit. But then at some point we made it, we got through the thing and we got to our sampling site. This is an overview of La Playa, and that's the Monte Cristo River. This is where we were. And this is a little bit of our, days in the field. We got to sleep in our very comfortable tents in a very cold temperature during the nights. And I think it was not too hot during the days, but a little warm for Humboldt patterns. And then these people, some of those were like the people that were with us during the whole time. We had a guy like doing cooking for us, which was like, oh my God, saved our lives. And then 3 other people that were like supporting us during the whole time there. And in addition to that, we also have local community carriers to help bringing things in. And then when we were leaving, bringing things out. And this was our not too fancy lab and, kitchen plus living room and everything that we had during those like 2 weeks, 20 days that we were there. We got to see many, many nice animals and plants. Not gonna leave that out, but I am a little biased on the animals. And then just some illustrated here. Pedro will know more about these guys, but I know that these guys is still unnamed. It's a new species. This frog was named right before we stepped into the park. So it was like something new that got a name just on that year. And the toucanet shown there is, I think, one of the rarest and possibly the most endangered toucan in the world. So we got to see really, really special animals. The Andean bear was really like around our camping site. That was really happy. We got some really nice views and some behind the scenes, how he got the shot. I always say don't do that. But But it was like, I know it looks like really like, why you would do that? But the guy, in this case, the bear, was coming, just like really curious. And we had a team of people, also a Parker range with us. So it was like as safe as it could be. But that's how we got it. And, you know, the not so good part of having the bear around, the bear found our small mammals traps, and Silvia wasn't so happy after 3 days. The first day was crying, oh, the Andean bear. And then the second day was also great. And then the third day, our traps were damaged. So I was like, can you please not do that? So it still worked. We still got working traps, but it was a downside. So anyways, going a little bit on the mammal side now, these are some of the species that we have observed and collected during this expedition. We got a really, really high diversity sampled during our 15 days of opened traps. And most of the mammals that we have collected there, they are actually apparently unnamed. This was the first published work based on the genetic data of that material. And based on genetic data, we could not match the mammals that we found there with anything else that is known by science for over half of the species that we have collected. So from the 16 species that we have seen there, we only could find matches for 4 species. That means majority of species from that site are not named. And it really stressed the importance of preserving that area. The 2 highlighted species are 2 that I'm gonna talk a little bit more about, because after knowing genetic data of these mammals, now as a systematist, taxonomist, I'm gonna get to look at them, the morphology of them, to give them formal names. So they are known by science. And while we don't have names for mammals or any species, we cannot work to preserve them, right? So it's important and really urgent that we get to look more deeply to each one of those cases and decide if they really need to be named as new species or not. So you might have seen maybe Marmosa chachapoia was the very first one named. It's an opossum that we collected there. So this species was published as new species last year. The 2024 is a typo. It was 2025. And it made, locally, it was a big hit. The media was putting in everywhere. So this little guy got a little bit famous. Another species that is also new that I already could work with the morphology and confirm the uniqueness is this little semi-aquatic rodent. These guys are really special. They are part of a tribe that live close to water, and they are relatively rare. You never collect too many of them. We have collected 1. And the publication for the new guy, it's already accepted. So it's probably gonna be out soon, within a month.
So this was probably the first expedition where I went as a full-time photographer. We had a herpetology team, so I was not the herpetologist for the trip. I was a photographer just taking photos. And the team was very excited about this rodent, which I was not sad about. Because it's not the most beautiful of the things that they collected. It's probably the ugliest, I think. But they were very excited. So they were pretty sure it was a new species. So we did take some additional time to have some of these pictures taken in the field. So we have fresh photos of it before they're made into skins for the museum. So the plates are better. So these are all from the very fresh animal. And because we didn't have a lab and I didn't have all of the settings, so this is some of the adaptations that we had to make to take those photos right there. So this is the background. This is the animal right there. Just me and taking that shot. Another thing that we have there, and the way I took most of those photos, because a lot of those animals we did not see in the wild. They were just like collected in traps and then brought alive to the camp. So one of the ways I had to take pictures of them without them escaping is that we have set up a tent just for photography of especially small mammals. So I was inside that tent for hours and hours and hours and taking photos of one mammal and then waiting there for till the next mammal would come, they would cross. I was just staying there. And as the days progressed, the tent smelled more and more like rats, rat piss, and all of those things, and sweat and all that. And I was there, and these two ladies were there helping me a lot. So this is, I was taking the photo pretty much from here. So this is my view of it. And this is a black background, and I do like flash photography and play with the light, so I can take some of these pictures of those animals. So these are all those animals that are probably unnamed to science. Those pictures are cute, they didn't make the paper. And this is the famous Sequoia.
Squirrel.
The squirrel was there. That's the only single shot I have of that animal, and we did not collect the squirrel, but I did see it. Me and this other one guy, of course, the last day of the trip. So I was waking up every single day at 4:30, 5 a.m., crossing the river, staying there at the place where the park ranger said, I've seen this animal here a few years ago. So I was just sitting there looking up a tree for days and days and days, and it wasn't showing up. So the last few days, I was like, this squirrel is not going to show up. So I went to take pictures of that toucan. So I had my camera all set up for a different area, different lighting, and somebody shouted, the squirrel is here. So I ran and took quick pictures. I had like probably 30 seconds to take these pictures and just vanished. So I have this one blurry, bad shot of the squirrel because I wasn't ready, because I was doing something else. I was hired for one job. Not do it properly. But still, it's the only shot of that species alive, and it's probably a new species. So we work with squirrels, so we publish text with National Geographic with some of my photos and my text explaining what her work is with squirrels and talking about the diversity. And this was the photo that she showed in her profile. And this is another. Behind the scenes, so for every photo like this, there are like dozens of.
Especially because it wasn't feeling.
Yeah, that's true. So the last day we were leaving the field site, the team got hungry and they went to buy street food, and they got this chicken with mayonnaise, and I said, you shouldn't eat that. I was the only one that did not eat it. Everybody else got really, really, really sick. And when we got back to Lima, we went to the museum and she had to look at the specimens, and she was really sick. And I still managed to get one of those shots. Never eat food like that. So this is our summary for that trip, and this is the second project that we're going to talk about. And this was fairly highly focused on storytelling, because I was more and more into doing like communication and broader impacts and outreach. So we put together a big team of biologists, of communicators, a designer that did all of these logos. And we did go to this incredible place in the middle of the Amazon. This is our arcing to get to the field station right there. So this is a very big field station built by the museum in Brazil, Museu Gildi, where we did our masters, and we still collaborate with the museum a lot. So we were there for about, what, 3 weeks, a month. in this field site with our team. And this was right after COVID, so it's like the field station was closed, staff was reduced, so it was like a really small community of people working together for almost a month on this project. This is our team, and as you can see, we have already added 2 new members. And one of the things that we discussed with the team before we even put the proposal together is that, okay, we can do this, we're in, but we are going to bring our kids, and this is non-negotiable. Are you okay with this? And the whole team was okay with it, and we understood that there were going to be challenges with having small children with us in the field, but the whole team embraced the fact. So this is the kids. How old are they here? So Leah's
2 and 4.
2 and 4. So this is in the field station, and this is in the river close by where we could see river dolphins, and these kids had a great time. And the rest of the team really embraced the fact that the kids were there. So we were taking turns. So who would stay with the kids? Sometimes we would bring them to the field with us. It was probably a very unique experience. You can ask them later if they remember some of these things. So this is probably his first contact with a snake. And one of the teams that were there, they were specialists in praying mantises. So they had this light trap collecting a bunch of insects. And one of the kids' favorite pastime was to go to the light trap and just see all of the insects that were there. No, I did. Did I ask too much? Information slide. Sorry.
Okay. So in this project, we were a little bit out of our more traditional place, which was like really focused on science, because we're like doing other stuff and working collaborative with the team that was focusing in other, right? Other than just like, which mammals live here, which herbs live here, which are more like our core. But we were still focusing on our core as part of the team. I was inventorying and like, you know, trying to figure out like which mammals live there. And Pedro was doing herbs, but we were doing some stuff different. And here is illustrated some of other team members working on like their time lapses. We were using kind of like new technologies to do investigate behavior of night creatures in the Amazon. So there were like UV lights to see like which animals would reflect UV versus not, you know, all sorts of like distinct stuff. So for mammals, we did a lot of the UV tests. This was one of the possums that were like, we could test for UV reflectance. And as you can see, the guy was really purple, which was nice. This was the most scientific portion that resulted from that expedition, which was a poster comparing and contrasting the color of the animals of the species that we collected during that expedition under UV light versus not UV light. So that's just like the most extreme example. Normal light and UV light, these opossum, Mammals ops woodley, it was really, really different. So there are like some hypotheses about like why animals would shine or not under UV. Nothing settled. There's a lot of like discussion if this is just a random result of some like properties of the chemicals that they have in their body, or if there is some selective pressure that have, you know, made them like that. And that's pretty much Pedro's turn, which is
And this is one of the challenges of having kids in the field. You know, everybody has to wait, and it's we're under their clock. So one of the focuses of this was storytelling, as I said, and we focused on night creatures. And one of my PhD students now, he was a master student back then, and his partner, which is a designer, they were invited to give a TED talk on the main plenary. And a lot of what their focus of their talk was in this, about this expedition. And so they were talking about the Amazon and the importance of conservation and the importance of studying biodiversity. They also highlighted our work with UV. And one of the things that we do have as a result for the UV project is that we have dozens of new records of UV fluorescence in many, many different animals, from onychophorans to a lot of insects and a lot of animals, vertebrates like snakes and a lot of mammals, as Silvia mentioned, and lizards and frogs. So we're only beginning to understand what UV does and what it means and why it evolved and what are the reasons for it. But this was highlighted and it's been seen by over 300,000 people, this talk. The other thing that we did together is we put together a exhibit to the museum in Brazil, Museu Gildi. So they were renovating and they built an immersive exhibit. So this is within a closed space with 4 walls and we have 4 different projections and sound. And I'm going to try to mimic this here. It's not going to be the same. And I do hope it works because the resolution was not ideal for the computer, but try to imagine being in a room enclosed by all this video and sound. And this is what this exhibit is. This exhibit was supposed to be there for only 6 months, and it's been there for 3 years, I think, because it's very, very popular and it's been visited for 500,000 people a year.
Uh-oh.
It's coming, it's coming.
Ish.
It's glitchy, but you can get the idea. Very glitchy. I think you're gonna have to go to Brazil to see it.
There's so many animals shown after.
I can go away, go away.
It's a combination.
I see, right, that they compiled to show.
Okay. He can take it. Let's just think.
Go to Brazil. I'm pretty sure that's gonna be there for at least 1 more year. Which brings us to our last project, and this is a current project that started after we got here. So the idea for this really stems from something that I was thinking about for a long, long time while I was doing my postdoc in Brazil. And then Silvia got involved with the project and we submitted to a couple of different agencies and it got rejected and we kept improving. And then we had kind of given up on the idea and then we moved here and she wanted to apply for an NSF grant. And I said, why don't we just like get that idea and revive it, maybe change a few things and resubmit it. So it got funded by NSF. And the idea behind it is that we're going to try to understand some of the aspects of diversification and evolution of biodiversity in the Amazon. And for a little bit of scientific context to this, people have been talking about diversity and endemism in the Amazon for a long, long, long time. So this is a paper by Wallace from 1852. And for those of you that do not know Wallace, he was like the co-discoverer of the theory of evolution. So Darwin got very, very famous because of it, but they have published papers together before on this idea. But Darwin had a bigger book with more elaborate ideas, but Russell Wallace was thinking about the same principles of natural selection and evolution. And he was in the Amazon a long, long time ago, and he was seeing all these monkeys that have had different distributions, and he was trying to look at insects too that had these like similar patterns. And he saw that these monkeys, different monkey species, were not getting together and those separated by big rivers. So he was one of the first people to observe these and pose some questions like, why are some species ranges delimited by rivers? And then this is a rough map of the northern South America, and these are the main rivers that are there. And a lot of people have worked on this, and one of the main proponents of the areas of endemism in the Amazon was Joel Craycraft, based on a really, really big data set of birds. Joel Craycraft happened to be in my PhD committee, and he was trying to push me to do a lot of biogeography. Even though I wasn't really interested in biogeography at the time, I was doing courses like evolution of reproductive modes and systematics, but he like planted that seed there. And then when we moved back to Brazil for my postdoc, and we were trying to think about different things, we were trying to understand those things. So this is what he proposed, that some of the major rivers had separated so many species that some of these areas could be considered areas of endemism. So he proposed some of these areas, and this is a very well-known subject for anybody that works in the Amazon, so the Amazonian areas of endemism. So these are the major areas of endemism, but people have played with it and tried to further split these areas, and there's a lot going on. But I want you to focus on this image and the next, on that circle over there. That's the island of Marajó.
Just make a note. What is endemism?
Endemism, I should explain what endemism is. It's a species is only known from that single area. So we have plenty of species of salamanders, for example, that only occur in northwestern United States. So that means that they're endemic from northwestern United States. Some species of lemurs only occur in Madagascar, so they're endemic from there. So what this means is that, from Craycraft's perspective, that some birds species are limited to those areas within those rivers. So that's why he delimited those areas of endemism. People in the Amazon and that work with biogeography are obsessed about these areas of endemism and testing the hypothesis that rivers really separate species. So these are just some examples. This is a study with birds, and they confirm some of these areas of endemism for birds. This is another paper for birds. As you can see, that tiny white area is not in any of those areas of endemism, and it usually doesn't have any samples, so they can't really test it. The same thing here for avian parasites and bats. They discuss the areas of endemism. So these are their sampling, and this is sampling, and that is again blank. No area of endemism delimited for Marajó. So what's special about that place? So that white blank area is this, the island of Marajó. So this is the Amazon River, and these are other rivers that really isolate Marajó from the continent, which makes it the largest fluvial island on the planet. And that area is so big that it's about 3.8 times the size of Humboldt County. That's how big that island is. And there are several interesting aspects about it, including that it's adjacent to 3 different areas of endemism. So Guyana right there and 2 different areas here. They have a very marked east to west gradient with like a more forested area to the west and a lot more like open grassland vegetation to the east. And it's very, very, very poorly studied. There are no species lists available for amphibians, for reptiles, for mammals, for insects. It's really hard to get samples, and that's why it's not really often included in any studies. So I began to discuss with my supervisor for my postdoc at the time, where does this fauna come from? Is Marajó its own area of endemism, or it should be connected to one of the three that's adjacent? And he's a bird person, so he had a firm answer that it should be connected to one of these three. But as I looked at the literature, that's not really the case, and our preliminary data was showing something different. So that's sort of how we framed the project. We want to know, first of all, what species are there, if there is any difference from the western, more forested area to the east, and where did these animals come from, and where does Marajó stand in terms of areas of endemism in the Amazon? So this is our, what we did. This is the project that was funded. It includes fieldwork, and this is the trip that we took last year. So we flew from Humboldt to Belém, which is already a long flight. You can how long those flights are. And then we took a boat from Belém all the way to the right in the middle of that forested area. And this trip takes roughly 24 hours in a boat to complete. 28 hours. We got to our sampling site, and then we were in a hotel there, sampling around there, and go all the way back again.
Okay, so you got an idea about the project, and I get to show the most interesting part, which are photos of us in the field. So this, as Pedro was showing here, Anajás, that's the city that we have, where we have done our field work. And this is an overview of, the Anajás area when we're getting there. And me working in the field. So this project, although I am like one of the heads of this project, I have other people. Pedro is a co-PI, and I have also another researcher that is another co-PI from Angelo State University in Texas. And we do have several collaborators from 4 different institutions in northern and southeastern Brazil. So it's a really like collaborative team. Here we have Edson, which is a co-PI, training his student Rachel that joined the team. Rachel is an undergrad from Angelo State University as well. She was learning how to set traps to capture small mammals. Here we have another collaborator from southeastern Brazil, from USP, Marcelo, handling a snake, and 2 grad students from Brazil that work directly with him and related to this project as well, learning sampling techniques. Another student, this is Cal Poly student, undergrad student at that point. Now she graduated, Hannah With. So she also joined, and I was like teaching her also sampling techniques. Another grad student from Brazil, recording some frogs, I think. Yes, so doing some sampling for frogs. Another very important team member, David, which also joined, and Leah was also there. Slightly biased toward frogs. I don't approve that. I'm kidding. I like that. And the team working in our improvised lab, which is really, really fancy compared to the Abseil lab that I shown, right? So this was a room that we organized to be processing the samples that we were bringing from the field. And we have 1 master, 1 PhD student, and another master student, all from Brazil, 2 of them from southeastern and 1 from northern Brazil, working on the herpetology group, like frogs. They were fixing frogs at this point. And here is a little, illustration of some of the animals that we got to see during this expedition. We got several new records of vertebrates that had never been registered before at Marajó. And that include, for example, I'm going to go with the salamander, which that's the Brazilian salamander that has not been known from the island. So it's a first record in the island. We also got some mammals that were not sampled there before. And I think there is a new, Pedro, no, a frog? New frog or new lizard?
Potentially a new snake, but we only had one specimen. It's a very complicated group of snakes. So even though we're 99.9% sure it's a new species, we're probably not gonna work with it for a while. It's too hard.
Yeah. Well, okay, but anyways, showing you a little bit of the nice images, all Pedro's photos. And just to, if you were wondering, like, this is a project that we just started, right? So we don't have results to show you. We just have like our preliminary results, which was like checking references, and our own previous work tells us that the story of the origin of the Marajó Island vertebrates, it's not that simple. It's not that we're gonna find a single route coming from northwest or from south for our groups. We are expecting to actually find a diversity of explanations for this particular group entered these many million years ago through this route. So, you know, different explanations for different groups, probably. It's much more confusing than we could expect. And this is to illustrate a little bit of also like remind us that we are showing the fieldwork side of the projects, and none of those projects are composed solely of fieldwork. I wish sometimes, but because fieldwork is so fun, when we come back from the field, we discover we have way more work to do than the field work per se, right? We have lab work to do, we have museum work to do with those animals. So for the Marajó project, just to give a context, we have the intention, we are using both samples that we have collected in the field. We are also using museum specimens samples, which we call historical samples, because they have not been preserved for genetic studies properly. And in order to access these type of samples and put them together with the modern samples to use to address the questions that we have and generate genomic data for these samples, we needed to establish a historic DNA lab, which was done, and it's now working. So the first 1.5 years of the project, I was working here to get this lab, you know, established, all the equipment needed, and we have already like done some extractions from these historic samples and send out for sequencing. So we are working, generating genetic data at this point. We don't have the data back yet. We are still working on field surveys in Eastern Marajó because the photos I showed you were from the western portion of the island, which is the forest area. Now we're working on the grassland area to get samples and compare, you know, one with the other. We are also starting to work on our, like, what are going to be our phylogenetic analysis and computer side of the project, and trying to generate more and more training opportunity for students, which is now, after the field work is done, pretty much the lab work and the bioinformatics work. So that's just like to wrap up where the Marajó project is at this point. And I think that's it. With that, I would like to thank you all for your attention. We would like to thank you. And also, we are very grateful to all the people that joined us in the field, collaborators, students, all the institutions, including museums and universities that have always supported our projects, and the funding institutions, right, that make all this possible. And if we do have time, which I think we do have a few minutes, we will be happy to get questions from you, to answer questions. We have this video, which hopefully will... That's a video with some images from the Abyssal project, which are really cool, in my opinion. I'm biased completely. So if you have questions, maybe get it.
Yes. It's
Put a little more light on.
Yeah.
I
see. So.
This is the Mammals and Sequoia, the one published last year. Yeah, just so it's a little like. No,
chill.
this guy was particularly. These, but that's not.
Especially the rodents.
Yeah, yes.
The rodents are just like gone.
Sure will always help.
He never gets to explain, but
I know, right?
it's set intent.
Yeah.
Just, explain that.
Yeah, so if they didn't escape.
Question?
This is kind of for most, is there like, I mean, you guys like gone like a lot of like northern South America, is there any places that you like really like to go to like in the future and research like certain like animals that like can pique your interest? Just the region to see like what.
Do I have to cite just one? Well, I am sure there are many. I would say I'm very, very curious to visit the point where Venezuela, Colombia, and Brazil meet, where we have the tepuis. I've never been there. I think he visited the tepuis before or close by. Yeah, I'm really, really like eager to go there. Just to select one. I have many.
Anytime we think of projects like that, we sit down and open a map, say, I want to go here, I want to go there. And we've done that sometimes. I want to go there, and we're just so obsessed with that area that we managed to get there. Kashwana was like that. Where can we do this project? And we said, we want to go there. They have a field station. She's never been there, and I've been there. So it's amazing. It's going to be good for the kids. It's going to be good for the project. We should go there. And I would say there's this one place in the border of Brazil and Guyana called the Tumucumaki Mountains. That's where I want to go. Because it's been sampled, but not well enough. And most of the specimens are in a collection that's not really accessible, and genetic samples are not that great. So I would really want to go there. So it's almost like that general area with a lot of highlands and lowlands. So a lot of endemism species that are probably just there. It's probably full of new species, but it's also really expensive to go there. And the areas where I want to go is like only by helicopter and things like that.
I know the study's still ongoing, but do you guys have any ideas towards which area of endemism the Marjo Island was a part of?
Do you wanna answer or I answer?
So do you have the presentation? Because I can show the map. But the bird folks are really convinced that is one of the southwestern area. But when we look at preliminary data from other groups, that's not always true. But I thought initially that it was this. So that's the island, right? And it's connected to the Guyana area of endemism, then Belém, Xingu, and to the top, which are also over here. So bird people always think it's these two. Sometimes they're
The Tapajós or Xingu.
Yeah, but we do have some evidence of at least some groups that form a clade together with the Belém area of endemism. And although the Guyana shield is older and less likely, it seems there are groups that could be linked to the Guyana as well. But I think the majority of them are going to be Tapajós Xingu, but it could really any.
I have, just like in previous studies that I did with the mammals, I have had samples from Marajó for two different mammals. One was a squirrel, the other was a opossum, and the squirrel closest link was with the Guyana, which is the snort, and the opossum was
It's the same.
okay, was with the Xingu. So That's why I think it's not gonna be super straightforward.
Yeah, for
One.
most of the birds, it seems to be the top and genes, but for most of the birds, it seems like that connection is younger. Also, and like, I don't know, we don't know. Yeah, I wasn't convinced by the answer from the bird people that, oh, it's very clear that, but some of them still say, you don't need to do that project, because we know it's from the. I don't know.
So there were a group of pictures of various animals, I think it was just after this.
Towards the end.
Towards the end, yeah.
Yeah, that's the Marajó project collection pictures.
Yes, that one, so lower left. It's just a leaf.
It's not a leaf.
Oh.
Pipa, right?
This is a pipa, it's a pipid frog, they are very weird, unique frogs. But they're one of the most ancient clades of frogs, and even though they're weird, they've looked like that for over 100 million years, so some of the fossils, they look just like that. Pretty much, but they're aquatic, they do come on land sometimes, but they are aquatic, and they have these bizarre reproductive modes. And if you Google pipa reproduction, like the females will have their eggs on their backs, and the tadpoles sometimes develop there, so the young froglets would just like crawl out of the skin. It's pretty amazing. Suriname frogs, yeah. Suriname frogs, Suriname toads, yeah. He knows.
How do you take a chromosome sample to know what do you take to extracting?
For like to do genetic studies?
Well, to find out what's a unique species, you have to take a sample.
Yes. We collect a little piece of tissue from like, it could be like several different types of parts of the body. It could be like a piece of liver or a piece of muscle or a little piece of the ear. And then you submit this tissue sample to an extraction protocol, and then you do PCRs and whatever other lab protocols you need to get DNA sequences out of that, or to sequence the genomes of the animals, and then you get to compare the genomes, the DNA sequences of those with whoever is already known by science. So you can see like how different it is.
You're not killing them.
So for us to inventory an area and to set up like who is in here, we need to take voucher, we call voucher specimens, which is just like a representation of the biodiversity. But we do that under like lots of permits and using specific techniques for the euthanasia of these animals. And we take just a few specimens, sometimes just one per species, in order to have a representation of that biodiversity in that time and location. Yeah.
And so now you're saying Humboldt has its own DNA lab.
Well, yes, we already had DNA labs before here at Humboldt, but not a historic DNA or ancient DNA lab, which has specific conditions to avoid external contamination. There's a lot of extra things you need to be worried with when you like trying to extract DNA from a sample that has been living in a museum collection for 200 years, for example. There are like many possible contaminants that went there. You may be sequencing like fungus, bacteria, other hands of other people, you know. So it's a lot of caution that you need to have trust.
Ideally, we take fresh samples, but if you cannot have fresh samples, you can go to collections, and sometimes it's just taking samples from specimens that are over 100 years old. And it's easier with skins, so mammals and birds, but for amphibians and reptiles, we're usually preserving formalin. And formalin degrades DNA and does weird things with DNA. But there's been a lot of research into this, and some people are managing to get some DNA, not high quality, but enough DNA that is useful, so we can use those old samples from collections to use as well. We're not doing this for this project because that's a whole different protocol and a whole different beast than just like skins. Formalin is a whole thing.
but just using this question to also like deliver another message. Like it's also showing like how important it is to have the museum specimens, because we now have the technology to access DNA from museum specimens that have been in museums for years, from species that are many times gone, their environments are gone, and we have a lapse of what was there at a given point. So we can study retrospectively what we had at some point. So it's important to keep collecting new areas that have not been surveyed. And this is why we take some specimens.
So by accident, I'm just, sorry. We're on the right slide here, because this is like a process. So we collect the animals and I photograph them. So that student right there is killing some of the animals with the whatever she's using. I don't remember the name of the chemical, and then giving it to Giselle, and she's the one taking a tissue sample and putting those vials and then preserve it. And then Luana is preserving them in formalin. But back in the day, people were not doing this. They were not taking this. They were skipping this step. They were going straight from this to preserving. And once in formalin, it's really hard to get the data. So now we do that extra step to take a tissue sample from every single specimen. Right? So we do have the genetic resources.
Just trying to imagine, you must feel some kind of pressure, like you're the first person to like try to identify a species and sell it, maybe, or at least do your best to describe it, the planet. Maybe hope other people will come and study it or whatever. What does that look like? What can you try to ascertain, or what do you have to kind of leave to the next people? Especially, I think in some cases you're saying you're really just getting the skins and the rest of the animal is leaving behind. But certainly there's all kind of glands and other pieces you're leaving behind that might really be informative.
You can answer, but I have a question for everybody before answering that question. So what do you think is the average time between this, let's say they collected a thing that's potentially new, to the actual description of the paper? What do you think the average time?
5 years.
5?
5 is the minimum.
But it's time. 10 years or less. Raise your hand.
And the last.
10 years or more? 20 years or more?
Oh no, where are we going? Yes.
The average time for all taxa, including mammals and reptiles, is like 20 years.
Oh no, yeah.
The first available specimen of that new species to like formal description was that, like an average of.
So before we forget to answer to the question.
Yeah.
If I got your question, like if I understand it, if not, you tell me. But you know, like the work of describing and naming a species is the very first step for everything else that needs to happen. So it's a really, although it's like in some areas it's really like neglected, and taxonomy, it's not a field that is like, especially here in US where most diversity is already described, it's not like highly you know, like, oh, that's what I want to do. But we feel like, especially coming from those areas that are really neglected by science and where diversity is so high and we need so much attention still, this is like the very basic step for everything else to happen. So if you ever want to get to the conservation of that, you actually need to usually focus on describing species and naming species. So I think it's a very important piece that hopefully I'm contributing with and other taxonomists. So we need more taxonomists, basically, more explorers, right? Because 20 years is non-acceptable, right? But that's the reality. And for Marmosa Sequoia, for example, that I have shown, we named it last year, 2025. We were in the expedition in 2018, and that's 7 years. Why it took so long? But that's a lot of things that happen in between, like, okay, in the field, when I collected that guy, because I work mostly with marsupials, I looked at that species and was like, that's a new species. That's different. I knew that, but then I had to study genetics, then the morphology. To study the morphology and then anatomy, I have to compare with specimens that are living in many museums around the world. So I have to travel, collect data. compare. So it takes really a long time to make a very solid work and avoid naming species that are already named, right? So because you don't want to do that. You want just to name who needs a name. Yeah. I never went to it. Okay. Well, I haven't done that yet. Yeah, okay.
We do have to cut it off.
Okay, sorry.
And you're welcome to keep asking, but I just want to let everyone else go if they would like to, if people still want to ask questions and you're okay with that.
Yeah.
That's fine, but I do want people to feel released.
Yeah, yeah.
But please come back. February 18th is our next lecture and it's about the California beaver restoration program. So we'll